RNA analysis already, particularly from Andy Clark in the first session, and from Rafa as well. And now we have a whole session to tell you about 3 different projects that are going on or are just coming to completion in RNA sequencing, and the first speaker is Joe Pickrell from the University of Chicago.
Great, thank you.
So thanks to the organizers for inviting me out, and thanks to you all for getting up so early to come. My name is Joe Pickrell. I'm a student at the University of Chicago in Jonathan Pritchard's lab, and I'm going to be talking to you today about some of our work in understanding mechanisms underlying human gene expression variation with RNA sequencing. And what do I mean by that? What I mean is that if we were to take a sample of individuals, such as yourselves, and take any individual tissue and assay gene expression in a whole sample of individuals, what we see is that for any given gene, there's going to be some variation. Some genes are going to be highly expressed, or some individuals are going to have high expression of a gene, some individuals low expression of a gene, and some in the middle. And we also know that there's genetic variation in the population. We've seen many talks on identifying genetic variation. And so some of these genetic variants actually cause these differences in expression across individuals. So there are genetic differences segregating in a population which cause differences in gene expression. And we know that this genetic variation among individuals in gene regulation is important. I don't have to tell this audience, but I will anyways. For one, it's important in evolutionary genetics. What I'm pointing out here is the allele frequency of a particular SNP near the Duffy locus. And red is high frequency, white is low frequency. You can sort of make out the map here. This allele is at high frequency in Africa and essentially absent in the rest of the world. And what this SNP does is disrupt a binding site for a particular transcription factor in front of the Duffy gene. The Duffy locus doesn't, the Duffy gene is not transcribed, the protein is not made, and it's thought that the lack of production of this protein is related to natural selection for resistance to malaria. And so genetic variation in this transcription factor binding site is important in evolutionary genetics. And as we all know, that in medical genetics, recent genome-wide association studies are identifying hundreds of genetic variants that aren't in proteins that are presumably regulatory, that are important in disease risk, and we don't know how they act. And so if we want to go beyond single examples like this and sort of comprehensively understand how genetic variation influences phenotype, what we want to do is have a model system where we can understand on a genome-wide scale how genetic variation influences gene regulation. And I'll point out that gene regulation is, of course, more than just gene expression. It's a combination of a whole host of factors. So natural genetic variation could potentially influence a whole number of levels of gene regulation. It could influence transcription initiation, either through perhaps chromatin accessibility, transcription factor binding. There could be genetic variants that influence mRNA processing, for example, in splicing, or RNA editing, or polyadenylation, or export. There could be genetic variants that influence mRNA degradation, either through microRNA regulation or nonsense-mediated decay. And that's all putting aside the very important protein levels. So there could be genetic variation which influences how things are translated or where they're localized or what have you. And so what we're looking for is sort of a way to understand genetic variation at all these levels and how they work to create, to influence gene regulation. And so the model system that we've been working with are the HapMap cell lines. We're using these as a model system for understanding the genetics of gene regulation. So in this talk, it's either 69 or 75 cell lines, depending on the slide, derived from white blood cells of Nigerian individuals. These are EBV transformed cell lines. And of course, we have over 4 million single nucleotide polymorphisms genotyped thanks to the HapMap. And now, thanks to the 1,000 Genomes Project, we have essentially complete knowledge of the common polymorphisms in this population. And we and others are generating genomic data. So for example, mRNA expression data, DNA methylation data, histone mark data. And so our goal then is to find out how this genetic variation causes variation in in, for example, mRNA expression. Today I'm going to be talking about mRNA expression. As Matthew pointed out, previous studies along these lines have used microarrays to assay gene expression. We're going to be using RNA sequencing. So how does RNA sequencing work? Just a quick overview of how this works. We're going to take each cell line, isolate polyadenylated RNA, convert it to cDNA. We're going to fragment the cDNA and sequence short reads, and in our case, either 35 or 46 base pairs on the Illumina platform. We're going to count the reads that come from the exons of the gene. We're going to process it, and I'll get into a little bit more detail of our meat grinder here. And we're going to end up with an expression level for each gene or exon. And this is analogous sort of to a microarray, but using RNA sequencing. And so with this technology, what can we look at? We can look at essentially at this. We can look at steady state mRNA levels, which are sort of a balance between transcription initiation and mRNA degradation. And we can look also at variation in mRNA processing and splicing and polyadenylation and things along those lines. And so what am I going to tell you about? First, I'm going to tell you about how we're using these data to identify eQTLs, so variation in steady state expression levels. Second, I'm going to tell you about how we're using these data to identify genetic variants that influence splicing. And then I'm going to talk about some unpublished work that using this data to show that splicing is relatively error prone. And so I'll start with mapping eQTLs. And so I'll just sort of show sort of an outline of how we're doing the data analysis and give a couple caveats. So as I said before, we're using RNA sequencing. If we have, this is sort of a schematic of the genome. In blue here are exons, and here's a schematic of the RNA. The exons are all spliced together, and a poly A tail is added. And so, like I said before, we isolate these polyadenylated RNAs, convert it to cDNA, sequence short reads, and then map these reads to the genome. And so, a few things to notice: reads covering the poly A tail and splice junctions don't initially map, but we get these piles up of reads on the exons. So we're sequencing the exons mostly. And so this individual has one RNA, this individual has two, and we see twice as many reads piling up. Of course, it's not that simple. One complication is that read depth is influenced by GC content. And we've heard this several times about these technologies. So what I'm showing here is I've split all the exons in the genome into 200 bins based on GC content, and then I'm plotting, that's on the x-axis, these 200 bins based on mean GC content, and on the y-axis for one of these, this is just a lane of data, and on the y-axis is the relative expression of exons in that bin relative to the other lanes. So this is not within an individual, this is, these are biases that are happening across individuals. So in this lane, for example, the sequencer really liked low GC content exons relative to other individuals, to other lanes. And in this individual, the sequencer really didn't like low GC exons compared to other individuals. And so this is a somewhat subtle difference between what we've heard before, in that different lanes of the same individual will have different profiles of this, different relative expressions for the same exon. And so we can correct for this. If we just fit a little smooth line there, we can downweight exons here, upweight exons here, upweight exons there, and slightly downweight exons there. And we've sort of corrected for this GC content effect to some extent. And as we heard before, there are also a whole host of other problems. There are batch effects, technical variation between samples. So one thing we've found to be important is the concentration at which an RNA sample is sequenced. There are sex effects, and all these things we're not particularly interested in the sense that they're not going to help us identify eQTLs. And so to take care of this, what we do is calculate principal components on the matrix of genes by individuals, take the first 16 principal components in our case, and remove them from all the individuals. And this is similar to other methods that have been used in microarray data. And we think this seems to work relatively well. And so what we ended up doing, we count the number of reads per exon in each individual, correct for GC content and the total number of reads in a lane, quantile normalize these across individuals, so each gene now has the same normal distribution, and regress these expression levels on genotypes. And what are we looking for? Looking for something like this. So on the x-axis here is position along the genome. On the y-axis is sort of base-by-base coverage from the RNA sequencing. And so at the bottom here, I'm plotting a gene model. In blue are the exons of this gene. In red are the introns of this gene. And in each of these plots, again, this is the position along the genome and the expression level of that base across the level of the genome. And so what I've done here is in blue are bases that cover exons, and in black are bases that do not cover exons, annotated exons. And what I've done here is average across individuals with particular genotypes. And so this is a SNP that lies somewhere upstream of the gene. This gene is transcribed on the minus strand. And so there are 18 individuals that have the GG genotype. And if we average their base-by-base expression levels, it looks something like this. We see the nice peaks covering the exons and absence elsewhere in the gene. And there are 16 individuals that have the AA genotype at this SNP, and you see essentially nothing. So, and the heterozygotes are in the middle. And so what we found here is a pretty strong correlation between a SNP and overall gene expression. And we find about 1,000 EQTLs at some FDR threshold, 10% in this case. And with this many EQTLs, we can start to ask sort of broader questions about where do EQTLs fall. And people have looked at this before, and we're just going to sort of confirm here that what I'm plotting here is from 80 kb upstream of a gene. This is sort of your average gene. Here's the transcription start site, transcription end site. And on the y-axis is the probability of a SNP that falls at that position to cause an EQTL for that gene. And so in blue, light blue, are bins that fall outside of the gene, and black are bins that fall inside of the gene. And what you see is that right at the transcription start site, there's a peak. So a SNP that falls at the transcription start site is more likely to influence transcription of that gene. And there's also an enrichment throughout the genic region. And so we see this, and this has been seen before in other studies. So this is more of a confirmation. And that's all I really want to say about EQTL mapping. I think we'll have additional talks about this later. For the rest of the talk, I'm going to be talking entirely about splicing. And we all know, of course, that about 40 years ago it was discovered that eukaryotic genes are split into pieces such that the protein coding parts are scattered throughout the genome and then spliced together. And we all know this, and it's part of standard molecular biology. We also know that this is a completely crazy way to organize a genome, that it is completely and utterly inefficient in the sense that if you take sort of a longish gene, RNA polymerase binds to the start, and by the time it gets to the end, it's been 30 minutes. And if all the exons were actually together, it would have taken about 30 seconds. So that takes energy, and it takes time to transcribe all these things that are eventually just completely spliced out. And so if you take at any individual time, the elongating polymerases in anyone's body, most of them are going along, using up energy, churning out complete useless junk. And so this raises a couple questions. One is how does the cell know what's junk and what's not junk? And this is one of major unsolved problems in molecular biology. How does the cell find 150 bases here, 150 bases here, and say these are the things that are important, and everything else in the middle is essentially not? And second, where do these introns come from, and what are the forces that influence their evolution? We have a fairly good idea where they come from, but the lengths and distributions of introns vary in many taxa, and it's not entirely understood why. What are the selective forces that influence intron evolution? And so I bring up these questions not because I'm going to have an answer to them, but because the sorts of genomic data that are being generated now have the potential to answer these questions. And so first I'm going to talk about variation across individuals in splicing. And how did we do this? Going back to our schematic diagram again, we have the DNA at the bottom, the RNA at the top here, and we have these reads covering it. We're going to map the reads to the genome again, and this time we're going to convert these reads into fractions. So 1/3 of the reads in this gene come from this exon, 1/3 of the reads in this gene come from this exon, and 1/3 come from this exon. And if we look at a different individual that doesn't have that last exon, 0% of the reads in this exon in this gene come from this exon, half here and half there. And so we're going to sort of use this as sort of a quantitative phenotype to map. It's not the ideal quantitative phenotype, but it seems to work. So just sort of the details on that, I guess I'll skip. We do the same sorts of corrections, and we find about 187 genes that contain genetic variants which influence splicing. What does that look like? It looks something like this. So on the x-axis again is position along the genome. On the y-axis is expression level. Here, this is for the last 2 exons of a particular gene. There are 3 different known transcripts. Again, the exons are in blue, the introns are in black. And again, in each panel is this expression profile for 3 different genotype classes. So there are 34 individuals with a GG at this SNP, and note that this SNP falls directly in the splice site for this last exon. So there are 34 individuals that have an intact 3' splice site, and you see the exon here and an exon here. There are 6 individuals which have a disrupted splice site, and what you'll notice is that the level of this last exon is much lower than the level of this exon. And there also appears to be something funny going on in the intron, and that this is unannotated transcription. So the first thing we wanted to do, taking this example as sort of a starting point, is that we wanted to ask, are SNPs and splice sites generally enriched for splicing QTLs? This would seem sort of obvious, but previous studies using exon microarrays found no such enrichment, and there was some speculation that this is due to conditional on a SNP reaching high frequency, it doesn't affect splicing. There was some speculation of that. And so we wanted to ask, in our data, are SNPs that fall in splice sites generally enriched for things that influence splicing? And what we're going to do is use a hierarchical model, and it's sort of a mixture model. We assume there are 2 classes of genes. One class of genes where there's no such SNP that influences the splicing of any exon, and another class of genes where there's one such SNP. There's a single SNP that influences the splicing of a single exon. And we can model the prior probability that any SNP is an SQTL for a particular exon of a particular gene. So the probability that SNP K is a splicing QTL for exon J of gene I is modeled like this. And the important thing is that it depends on a parameter lambda, which is the annotation. So we can modulate the prior probability that a SNP influences the splicing of an exon based on an annotation, say, is it in the splice site, or is it in the exon itself, or does it fall in some RNA binding site? And then the question is, in this model, is the annotation effect significantly different from 0? And so we can test a bunch of different annotations. Here's what it looks like. I've just arranged a few annotations we looked at, and this is the log odds of a SNP in any of these annotations to impact splicing relative to those in non-splice site introns. And what you see, this is a splice site. Splice sites are definitely above 0. SNPs and splice sites are massively enriched for things that influence splicing, as are things that fall in the exon itself, indicative of sort of exonic splicing enhancers and things along those lines that are being disrupted. Things in other exons don't seem to really influence the splicing of an exon, and things that aren't genic. And so we can then go back to this example and say, yes, SNPs and splice sites are generally enriched for splicing QTLs. And I want to point out another interesting thing that I alluded to earlier about this plot is that there's this, in the 6 individuals that have a disrupted splice site, there's this bit of extra transcription. So can we definitively show what the transcripts are in this gene? We've relied up to now entirely on the existing gene annotations. And so what we're going to do, moving back to our schematic again, is use these things that didn't map anywhere. So there are these reads that cover splice junctions that don't map anywhere in the genome. We're going to take the first and last 20 bases of those reads, and we're going to identify splice junctions de novo. So we can just take, this is pretty simple, just take the first and last 20 base pairs of each read, map them independently, and see if you can find splice junctions. And so if we return to this example, we identify the splice junctions, and what do we find? So we find in these individuals, in gray are splice junctions that are known, and in red, which really doesn't show up, are those that are unknown, or unannotated rather. And what we find is that in these 6 individuals, there's a new transcript. The splice site is here to here. It's in frame with the rest of the protein, and it adds about 260 additional amino acids to the protein. And so we're getting base by base resolution of where splicing is occurring. And so in the last couple minutes, I just want to use that information to show you evidence that splicing is error prone. So just sort of the details of what we did there. We started with 1.5 billion reads. Most of them map directly to the genome, but then we can take the ones that don't and identify splice junctions, and we get about 400,000 putative splice junctions from 40 million reads. And what does an individual gene look like? Looks something like this. Again, x-axis is the position along the genome, y-axis here is expression level, and you see, and again here are the transcripts of this gene. We see the peaks of expression in the exons. And what I'm showing in the middle here are the splice junctions we identified in this gene. And in black are splice junctions that are annotated, and in red are splice junctions that are unannotated. And most of them are red, of course, but I've ordered them so the ones at the top have the highest number of reads. So there are 5,000 reads supporting this splice junction, 2,000 supporting this splice junction, but at the bottom, this is 1, 1, 2. So there are only a couple reads showing evidence of splicing between these sort of novel exons and novel splice junctions. And one worry, of course, is that this is artifactual, but it's not. So we identify approximately 300,000 splice junctions at a low FDR. About half of them are unannotated, and most supported by a few reads. And so if we plot, what I'm plotting here is for all these splice junctions supported by 10 reads, what fraction of them are annotated in blue here? 60% are annotated. What fraction are in EST databases? Maybe 70%. But what fraction of them match the consensus splice site directly inside of the junction? And so we didn't use this information. We didn't use the fact that we know splicing is sequence specific to map the reads, so we can use that as sort of a confirmation. And nearly all of them match GTAG directly inside. So these are true outcomes of splicing, just low-level splicing, and nothing matches a control. And so are these functionally relevant isoforms? What we can do is take, if things are functionally relevant over a long period of time, they should be evolutionarily conserved. And so if we take the annotated splice junctions, and what I'm plotting here is distance from the 3' splice site in bases, and distance from the 5' splice site in bases, and I've taken all such annotated 3' and 5' splice sites, and I'm plotting the average conservation score from this model. And what you can see, and so high means more conserved, and the exons are conserved, and the 2 bases of the splice site are very highly conserved, as you'd expect. You can sort of see the 3' splice site, you can see the branch point, and other sorts of important sequence features that are relatively highly conserved. And the same thing for the 5' splice site. Exons are highly conserved, splice sites are highly conserved, introns are not conserved. So what if we look at these things that are not functionally annotated? We see that it's essentially flat, that the new low-level splice forms are essentially not conserved to any degree at all. Of course, this is conservation over a long period of time. We can use SNP data, so we can ask over the last 100,000 years or whatever, have these things become functionally important? And so again, annotated splice sites, this is SNP density now on the y-axis. And so within splice sites, there's a lack of SNPs, as you'd expect. Within exons, there's a relative lack of SNP relative to introns. Now we can ask the same thing, go to the other class of splice sites, and there's essentially nothing. So there's no real difference between, so these splice forms are essentially what we're going to call errors. they're splicing, splice factors are binding, things are getting spliced together, but they're essentially either neutral or slightly deleterious over evolutionary time. And why do we care about splicing error rates? Well, we care because there's interesting theory about how the mutational target size for a regulatory system influences error rates, and how natural selection also influences that. And so we think we're able to measure now for one of the first times the splicing error rate on a genome-wide level. And we can look at things that correlate with splicing error rate, what I'm plotting here. So to measure an error rate, we consider a splice junction between two really highly conserved splice sites and ask what fraction of the splicing happens to non-conserved splice sites. And what I'm plotting here is I've split all the introns into bins based on their length, and then can sort of ask, are introns of shorter length, what's the error rate of introns that are short versus introns that are long? And what you see, well, overall there's a 0.7% error rate, which is relatively high, and short introns are more accurately spliced than long introns, as you might expect. There's also possible reasons to expect why highly expressed genes might be more effectively spliced. We do see that that's the case. On the x-axis now, I've binned genes according to their expression level, and we see a negative correlation. And this is, again, the error rate on the y-axis. So highly expressed genes have a lower error rate compared to lowly expressed genes. But we also know that highly expressed genes have shorter introns, so after correcting for that, we see essentially no correlation between gene expression level and splicing error rate. And that is all I'm going to say. I've showed you how we're mapping eQTLs with RNA sequencing. I've showed you some of the technical biases that we're dealing with, and I've shown you that most eQTLs fall near genes. We've talked about splicing QTLs, and so I've shown that splicing QTLs are enriched in splice sites and exons, and we can identify novel transcripts. And sort of where we're going with this is, can we identify precise binding sites in RNAs that are important for splicing? And I've shown you evidence that splicing is error-prone, that there's extensive unannotated unconserved splicing, that long introns have higher splicing rates, and we're interested in taking this to a number of species and a number of tissues to understand how these error rates evolve. And with that, I'll thank everybody who was involved, particularly the wet lab people, Yoav and Athma, who did all of the sequencing. And thank you for listening.
Thanks very much, Joe. Are there any questions?
Yep.
So the effect of these error splicings is fascinating. And suggests there might be quite strong selection against the splice signal in introns.
Mm-hmm.
Is that a feature you see over?
That is not a feature we see. and so a lot of this is confounded. Okay, so it is a feature we see, but we think it's effective GC content. And so highly expressed genes tend to fall in high GC content areas. The 3' splice site, for example, is very AT rich. Highly expressed genes have short introns, so the error rate is lower. So I think it's almost completely confounded with things like GC content and things along those lines. And the reason I think this, I guess, is that so we see the same correlation. So we see there's a reason I don't think this is the case, but I don't know.
You could look in polymorphism data for direct evidence for selection against
against splice site.
causing.
Yeah, that's a good idea. That's a very good idea. Yep.
So percent of error rate, if it's totally neutral and they have no effect, I guess that wouldn't be terrible. But if you create a poisonous product, of course, that's not a good thing. So I guess maybe it's a similar question to Gil's. I might imagine that these things are kind of popping up, being created, or maybe pseudo splice sites are being created, and then there's going to be a selection process on those. So if you look at the new novel exon product that's created in some sense. Are there any properties of that which are different from what you might expect from random? Are they shorter than random, or I'm not exactly sure what one might look for, but I'd imagine that there'd be some selection on what the novel product is.
Yes, I think that's almost certainly the case. But so we haven't been able to identify. So most of these things have one splice junction or two splice junctions, and oftentimes we don't know where the other splice junction is. So it's more difficult to know exactly what that entire exon is.
You know what, if you're coming along from 5' to 3', and you acquire a new 3' splice, for example, you know what you've shifted it to.
Yes,
and so I'm asking the new thing that you're making there, that the new coding product, does that look totally random compared to just any random start point in the genome, or can you see evidence that it's not random?
So I think there are two sorts of errors going on here. One is in exon recognition, so we're creating entirely new exons, and the other thing is sort of alternative 3' and 5' splice sites. And I think those are probably two different mechanisms, but we can show that for the latter case, where we have an exon here, and we find errors near that extend the exon by an amino acid or something like that, we see a fairly clear periodic pattern of where these errors are falling, in the sense that they tend to be multiples of 3, as you'd expect.
Maybe time for one more question while we, if there is one, while we switch.
You might also, I mean, nonsense-mediated decay,
Yes.
presumably.
Yeah. So that effect is sort of confounded between nonsense-mediated decay and actual selection against creating things that would then be degraded by nonsense-mediated decay.